Chapter 9, Part A, deals with the muscles and the muscle tissue. Obviously, you need to understand what the normal physiology is and the normal anatomy is for skeletal muscle, so that you'll be able to treat your patient when they come in with strained muscles and you understand why you're doing the different procedures that you are doing. Now, nearly over half the mass of the body is actually due to the muscle. What do the muscles do? Well, they take chemical energy in the form of ATP and they can basically transfer it to mechanical energy, allowing those muscles to contract and having force and causing movement. When we look at the muscles, we're going to look at different muscle tissue types. We're going to look at some of the characteristics of the muscle and then the function, of course. Keep in mind when you're looking at different books and reading different materials, some clues for you. If it says myo or mys or sarco, those prefixes all refer to muscles. So that's going to help at least get you in the right direction of what they're talking about. If you remember, there are 3 different types of muscle tissue. There's skeletal muscle, cardiac muscle, and smooth muscle. Now, in skeletal and smooth muscle, I know it gets a little bit confusing sometimes because the muscle cells, instead of calling them cells, we refer to them as muscle fibers. It's just something that, by tradition, that's the way we refer to them. Most of this chapter, we're going to be focusing on skeletal muscle. Skeletal muscle is packaged into the skeletal, the tissue is packaged into the muscles. The skeletal muscles are going to be attached to bones and or skin. Now, skeletal muscle fibers are very long. They're the longest of the muscle cells. They have stripes or striations to them. They are voluntary because you can control them consciously. You can decide whether you're going to move your leg or your arm, etc. When the skeletal muscle fibers contract, they do contract very quickly. So it's very rapid. It can be a very powerful contraction, but they're going to tire out fairly easy. It's kind of like, yes, we got the strength that we're going to be fast, but we don't have the ability to sustain it for long periods of time. Cardiac muscle is found only in the heart. We have a different chapter where we will focus just on looking at the heart. So we're not going to really focus on it much here, just comparison purposes, and that it is also striated. It is involuntary. You do not consciously control the contraction of the cardiac muscle. It's under its own control system, cells that we call pacemaker cells. The nervous system can have some influence on that, but it's actually the pacemaker cells that are controlling the rate. And then your smooth muscle is also involuntary. However, it is not striated. Smooth muscle is found in the walls of hollow organs, so the walls of your stomach, walls of your bladder, your airways, your blood vessels, posterior digestive system, etc. And this table just shows a comparison of the skeletal, cardiac, and smooth muscles, where it's found. You can see the histology slides here. Excuse me. You can see the striations right here with the skeletal, striations right here with the cardiac. These darker pink lines right here, those are called intercalated discs, which are unique to cardiac muscle. It's one way you can distinguish between cardiac and skeletal. Also, cardiac is branching, skeletal is not. And then your smooth doesn't have any of those striations. Some characteristics of muscle tissue, of all of them, is that there's 4 main characteristics. Things such as excitability. That means it's able to receive a stimulus and respond to that stimulus. Contractility is the ability to very forcibly contract or shorten when it receives that stimulus. Extensibility means it's able to be stretched out, and elasticity means it's able to recoil back to its original shape. So you can stretch it, you can contract it, and then afterwards, when it goes back to that resting state, it's going to go back to the resting length. That's very important because you don't want it to remain contracted. You don't want it to remain stretched out. What are some overall functions? It produces movement because it's attached, as we just said, to bone or skin. When that muscle contracts, it's going to cause movement. It helps to maintain your posture and body position, keeping your head up. If you didn't have any muscles at all, we'd just be kind of this heap with bones, and there's nothing to help keep that proper positioning. Muscles are very important in helping to stabilize joints. If any of you have ever had any, say, knee injuries, one of the things they're going to do in rehab, especially if you've had knee surgery, is to have you start doing exercises to help strengthen the muscles that extend around the knee to help stabilize that joint. Same thing with the shoulder. And when muscles contract, any type of movement like that will generate heat. So you might think, well, what does that have to do with anything? It helps to maintain that internal body temperature. Remember, if you get really cold, what do? You start to shiver. What is shivering? It's just muscle contraction generating that heat. There are some additional functions, things such as protecting some of the organs. Some of them do form valves, typically circular muscles that will open and close, whether it's allowing light into your eye or around your mouth, enabling you to open your mouth. And some of the other openings, like in the anus, so the bladders, is helping to regulate and control. Once again, all your systems have to work together. So skeletal muscle is an organ because remember, an organ is made up of at least two different tissue types. So you might be thinking, well, it has the skeletal muscle tissue, what else does it have? It also has nerves and it also has blood supplies going in there. Because muscles very actively contracting, etc., it needs to have very good supply of oxygen. It needs to have very good supply of nutrients. It also has connective tissue that's attached around the muscle and helps form some of those attachments, the tendons and ligaments. So each muscle receives a nerve, an artery, and a vein. Your skeletal muscles are going to be controlled by the nervous system versus like the cardiac muscle, which is not. As I just said, when those muscle fibers contract, it takes an awful lot of energy, it takes an awful lot of oxygen, and it's going to be generating a lot of waste products such as CO2 that needs to be, you have to get rid of those waste. You don't want them building up because it can build up to toxic levels. You have to get rid of it. So for that, you do need that extensive blood supply. As I said, connective tissue is going to be involved because you have these different layers of sheaths or coverings around the muscle. What are they there for? Well, they help to support the cells. They help to reinforce the entire muscle. There's three different layers: the epimysium, perimysium, and endomysium. Now, some of these prefixes may look kind of familiar to you, like the epi and the peri and the endo. When you see these, and you're going to see them again with the nervous system, epi is always going to be the outermost, peri is the middle, and then the endo is the innermost one. The endomysium is this connective sheath that's surrounding each individual muscle fiber. Remember, that's a muscle cell. Then you group those together, all of those muscle fibers together, and then you put a connective tissue wrap, if you will, around that, or a sheath around it, and that would be the Paramysium. And you call that group of muscle fibers a fascicle. Then you take several of these fascicles and group them together, and you wrap that with another additional outer layer, and that's the epimysium. And that's when now you're talking about the muscle itself. So in here, excuse me, here is your individual muscle fiber. These are the myofilaments. We're going to talk about this a little bit later. That allow for the contraction itself. Here's the nucleus of the cell. And so you wrap this with the endomysium. So this is a muscle fiber. Here's a muscle fiber. Here's a muscle fiber. Here's a muscle fiber. All of these are your muscle fibers. Each one is wrapped with the endomysium. Now you take this group and you wrap it with the paramysium. This whole group you refer to as the fascicle. So when you look at an entire muscle and you do a cross-section, you have all of these multiple fascicles in here, right? Each one has the paramysium around it. So you bundle all these fascicles together and now you wrap it with the epimysium. So just think endos, innermost, peris, middle, epis, outer. The muscles are going to typically span or move over a joint. They'll attach to bones. You're going to have muscles attached to bones in two different places because each end has to be attached to something. We refer to these attachment points as the insertion and the origin. So the point of insertion is going to be where the muscles attach to the bone that's going to be moving. The origin is attached to the immovable or the least movable bone. Now, those attachments can either be direct or indirect. If it's a direct, the epimysium kind of fuse with the periosteum of the bone or with the pericardium of the cartilage. If it's indirect, then you have connective tissue wrappings that will extend beyond the muscle as a tendon. Will you need to know the insertion and origin points of muscles? Yes, you will. We tend to focus on that a little bit more in the lab. For the way I teach the class, the lecture portion, we tend to look more at the physiology, and in the lab, we look more at the anatomy. When learning the muscles, there's different ways of doing it. Some people learn the muscle right then, learn the insertion and the origin and the type of movement. And that works for some people. For some people, that can be a bit overwhelming. Personally, for me, I think it's a little bit easier. And what I would recommend is as you're studying the muscles, excuse me, my recommendation is learn the muscles first, because it's not going to do you any good to know, oh, I can tell you what the insertion is and the origin, and I can tell you when this muscle contracts, which type of movement we're going to see, but I can't identify it. I don't know where it's at. I can tell you when the vastus lateralis contracts, this will be the type of movement. And I know that the vastus lateralis is somewhere in the thigh, but so are a lot of other bones. Can you distinguish it from the vastus medialis? Can you distinguish it from the biceps femoris? Can you distinguish it from the rectus femoris? So learn where the muscle is first. Can you identify it? Because my thought process, and you can take this or leave it, find what works for you. If you know where the muscle is, then you can look and say, okay, well, where is it attached. It has to be attached somewhere. You can pretty much figure that out. And then once you know where the muscle's attached, just look at it. If that muscle contracts, if it shortens, and you know where it's attached, then you can figure out what kind of movement you're going to find. So to me, learn the name of the muscle, be able to identify it, and then everything else just kind of falls into place. At least that's the plan. That's what we always hope for. So once again, this table is just looking at this structure and the organization of skeletal muscles. So we're looking at skeletal muscle here. So you can see here is the muscle we call the overall organ. The muscle fiber is the individual cell. So once again, the endomysin, you package all these individual cells in a bundle. You call it a fascicle, and it's wrapped by the Epimysium. Now you take lots of fascicles, wrap them together, and it's covered by the Epimysium. So skeletal muscle fibers tend to be very long. They do contain more than one nucleus. They're one of the few cells that has that. And yes, here's where we're going to, just add some more confusion to you. We're going to change some of the vocabulary. The plasma membrane, yes, it's a plasma membrane, but we're going to call it the sarcolemma. Remember I said sarco refers to muscle? Oh yeah, here we go. Sarcoplasm is the same thing as the cytoplasm. Why do we do this? I don't know how it actually started. There's a lot going on in the muscles you're going to find with muscle contraction, so it kind of distinguishes between some of it. I'm sure some of you are convinced that we have somebody in a little office who's like, how can we get the A&P students? Ooh, they don't have enough to study, so let's come up with some more vocabulary. Who knows? Myoglobulin is a compound that's found in muscle fibers, and it does bind oxygen. It's similar to hemoglobin that's found in the blood. So it does store oxygen, which is important because muscle contraction requires a lot of oxygen. There are some modified organelles, myofibrils, sarcoplasmic reticulum, and T-tubules. So we look at them, the myofibrils. A single muscle fiber can literally contain thousands of these. It's what most of the muscle cell is made up of. It does have different features to it. It's because of the myofibrils that you see the striations. You see that striping pattern. And they have what are known as sarcomeres, myofilaments, and we're going to look at all of these. So when we look at a muscle fiber, so this is the individual cell, the sarcolemma, that is the plasma membrane. Now, if you're thinking, well, wait a minute, what happened to the endomysin? It would be on top of or surrounding the sarcolemma. It's kind of like you've got a shirt on. That's the sarcolemma. And then the endomysin is going to be your jacket that you put, or a sweatshirt that you put over your shirt on there. So inside the muscle fibers, these are the nuclei. Like I said, there's several of them. And here you can see a little bit easier the striping pattern, those striations. So we call it the dark band and the light band. And inside the cell, here are some of the myofibrils. Now this is only showing some of them, but you could have thousands of these. Because you are making the muscle contract, which requires energy, that means you need to make a lot of ATP, which is made in the mitochondria. So the other thing we see is that muscle fibers tend to have a lot of mitochondria in them, which makes sense. So when we look at these striations, this striping pattern, it's very consistent in that you have these repeating dark and light bands. And of course, we're going to give them names. So the dark regions are known as the A bands, and the lighter regions are known as the I bands. And if you're trying to figure out, oh my goodness, how am I going to remember this? Dark has an A in it, and light has an I in it. For me, that's the easiest way of remembering. Within the A band, there's a darker A band, and in the middle of it, there's a lighter region, and that's known as the H zone. And then there's some protein that kind of bisects it in the middle that's known as the M line. In the I band, there's what's known as a Z disc in here. And we're going to look at some more of this in a moment. So right here you can see the dark band and the light band. The sarcomere. We're going to bring all this information in a minute. If you're sitting here thinking, oh my gosh, she's just throwing out all these different terms, how does this all make sense? It's all going to come together in a moment. The sarcomere, this is the smallest functional unit of the muscle fiber. It's going to contain an A band, and on either end of that A band you have half of an I band. So you have basically half an I band, an A band, half of the next I band. That's what the sarcomere is. So basically it's the area between those Z discs that I said was halfway through the I band. So you go from one Z disc to another Z disc. That is the sarcomere. That is a contractile unit. You're going to line all of these up. So here we go. The Z disc, it looks like a zigzag line. So here's a Z disc, and here's a Z disc. So from one Z disc to the other Z disc, this area right here is a sarcomere. There's going to be another Z disc over here. There'd be another sarcomere. So they're just lined up end to end. So you have half of an I band, the entire A band, and half of the next I band in terms of what the sarcomere is. Here's that H zone that I said was a lighter area in the middle of the A. And then right in the middle, it's a little bit lighter here, and right in the middle of that is this M line. What do the lines represent? If you notice here, it's kind of color-coded. You've got those thick redder lines and then the little thin light blue lines. Those correspond to the different filaments, thick and thin filaments. It's all very specifically arranged and regulated. It's not random. The actin myofilaments, those are the thin ones that look like that thin blue on that picture. They will extend across the I band and partway into the A band. They're going to be anchored to a Z disc. The myosin are the thick filaments. They look like that thick red on the picture. They are extending through the A band. So if you look at a picture of it, and thankfully they use the same colors, here's a Z line, here's a Z line. So here's your sarcomere. This would be the lighter area, this would be the darker area. So this would be the I, this would be the A band because it's darker. Within the A band, you have that H area, and then the very middle is the M. And what it is showing here, the reason why this is darker is because you've got those thick filaments, and for most of it, but not all of it, where it's the darkest area, you have both the thick and the thin filaments. In the I band, the reason it's lighter is because all you have are your thin filaments. That's it. Now in the H zone, the area in the middle of the A band, that's where you only have the thick filaments. So that's why it looks a little bit lighter, not as light as here, but a little bit lighter than the dark area here. The thick filaments are composed of a protein called myosin. You have these heavy chains and light chains that intertwine. The thing that you need to be mostly familiar with is that on the myosin there's a globular area that's we refer to as the head. And the head can bind to the thin filament. So this is on the myosin, which is the thick filament. That head can bind to a certain site on the thin filament, and when it binds and connects to it, that's what we call a cross bridge. And that is what's going to happen in order to get muscle contraction to occur. The thin filaments, they have the fibrous protein actin. Now, they also have some regulatory proteins that are bound to it. You don't have to worry about the G and the F actin. But you do have to be familiar with the tropomyosin and the troponin. These are the regulatory proteins that are bound to the actin. So over here is the thick filament, and all these little globules, those are the heads, the myosin heads. This is what is going to bind to the thin filament. So if you take one of those out, it's like having a big old handful of spaghetti or something. They almost look like twisters, the licorice twisters. So this is referred to as the tail. You put them all in a group like this, and each one has these heads. So it's right here that's going to bind to the actin, that thin filament. Here's your thin filament. Notice that you've got two filaments, and around it, this beige looking color is tropomyosin. Connected to that is troponin. Now, if you look at the individual actin, see these little couple like little dwellings? That's where the myosin head can bind. So that's the active site. So if that were exposed, the myosin head can bind to it. But notice the tropomyosin. It is lined up, so it is covering every last one of those. There's not a single one that's exposed. They're all covered up, which means if it's covered up, the myosin head cannot bind to it. So in order for muscle contraction to occur, the myosin head has to bind to these active sites on the actin, but they're covered up by the tropomyosin, which means you're going to have to move them. How are we going to move them? Well, that's where troponin is going to come involved. In order for muscle contraction to occur, you have to have the actin and the myosin bind together, but it can't happen because the active sites are covered up. So what's going to happen is calcium is going to bind to troponin. When calcium binds to troponin, all these yellow structures right here, what do I always tell you? When two things bind, you're going to have a shape change. So when calcium binds to troponin, it's going to cause a shape change, and it's going to shift this tropomyosin strand. It's going to literally move it so that then the active sites are exposed. Then the myosin heads can bind to it. But at rest, you don't want muscle contraction to occur, so you don't want those active sites exposed, so you cover them up. How? With the tropomyosin. And the troponin is going to help hold it into place. And this is a really cool picture of the myosin heads, the thick filaments, the thin filaments, and the cross bridging that you see occurring. There are some other proteins that are going to help with the structure of the myofibril elastic filament. There's some C proteins, so there's several different proteins involved. Know that it's not just one single thing. The sarcoplasmic reticulum. Now this is what we call the smooth endoplasmic reticulum in other cells, but in muscle cells they are specialized, so they are known as the sarcoplasmic reticulum. Associated with them at perpendicular cross channels, there's what we call terminal cisterns. The sarcoplasmic reticulum is going to basically store calcium. Calcium usually is not very high in the cytoplasm of cells. Usually it's higher outside the cell. However, in a muscle cell, the calcium levels are going to be low in the sarcoplasm, which is the cytoplasm, but it's going to be stored inside the cell. When you have excess calcium, it's going to store it, but it's going to store it in certain places. And where does it store it? It's going to store it in these terminal cisterns. The T-tubules are like these little channels or tubes that extend from the surface to the interior of the cell. So you have these little tubes from the surface extending in. And what we refer to as a triad is where you have a T-tubule, and on either side of it you're going to have the terminal cisterns, which is where the calcium is stored in the sarcoplasmic reticulum. So with the T-tubule, there are certain proteins associated there that are voltage sensors, so like voltage ion gates. And when they are triggered by a change in electrical current, it's going to trigger the opening of calcium channels in the cisterns, allowing the calcium to be released from the cistern into the sarcoplasm. So when that electrical charge comes down, which is going to be an action potential, when it comes down the T-tubule, it causes a shape change. It's going to ultimately trigger the release of calcium. So if you look at this picture, here is your muscle fiber, the individual cell. Remember, you have thousands of these myofibrils along here. So what happens is, from the surface area here, you have these, so here's the outer sarcolemma right here. You have these T-tubules, these indentations. So here's this outer surface. You have this T-tubules, this indentation, and it's going to move down so it can eventually basically pass by every one of these myofibrils. On either side, when we talk about the triad, you have the T-tubule, and on either side, this blue, that is going to be the terminal cistern. That's part of the sarcoplasmic reticulum. And in there is where you've got the calcium stored. So when you have an electrical impulse, electrical charge traveling down the surface of this muscle fiber, how are you going to get that charge to activate in here and do anything with the myofibrils in the interior portion of the cell? That's where you've got the T-tubule. So the charge comes down, whoop, it's going to move down that T-tubule. Same thing over here, you've got, it's going to move down. When it moves down, it's going to trigger those voltage gates. It's going to cause shape change and a charge change here along the terminal cistern, which is storing the calcium. That change is going to cause the release of calcium. Now it's going to penetrate in here right along with the myofibril, those where you've got the actin and the myosin, the thick and the thin bands. So contraction is when you activate those cross bridges. You cause that to form and you generate some force. Shortening occurs when that tension is generated by the cross bridges. And then when the cross bridge then relaxes and becomes inactive, then the contraction ends. The muscle's going to relax again. So if you look at those myofilaments, when the muscles relax, the thin and the thick filaments, the myosin and the actin, there's only a short area that they are overlapping. It's towards the end of the A bands. The sliding filament model for contraction basically states that during contraction, that area of overlap extends. So is your thick filament getting longer? Is your thin filament getting longer? No, it's not. It's just the overlap between a relaxed state. It's a very small, slight overlap, but when the muscle contracts, that overlap area gets much larger. So when the nervous system is going to stimulate the muscle fiber, the myosin heads are allowed now to bind to the actin, and they form a cross bridge. And that's going to start the contraction. When the cross bridge is attached, they're going to form. That myosin head is going to cock. It's going to release, and then it's going to bind to the next active site. Cock, push it, release, recock. We're going to see that. So it's what we call the sliding filament. You have the two fibers sliding basically past each other. So the I bands are going to shorten, the Z discs get closer as that contraction takes place. So here is a muscle that is relaxing. Notice the overlap right here. The blue is the actin or thin filament. The red is the myosin or the thick filament. The overlap is really minimal. When the muscle is contracting, look how much more overlap now you have on there. So in order for a skeletal muscle to contract, we're focusing just on the skeletal muscles. First thing you're going to have to do is have a nerve stimulation. Second step is going to be action potential, which is an electrical current. It has to be generated on the sarcolemma, the cell or plasma membrane of that muscle fiber. That action potential has to move. It can't stay in one spot. It has to move, or what we call propagate it, along the sarcolemma. And then you have to have levels of calcium inside the cell. They have to rise because that's going to trigger the contraction. So these 4 steps have to occur in a certain order before you ever even get to contraction. So steps 1 and 2, that nerve stimulation and the action potential being generated, is going to occur at the neuromuscular junction, where the nerve and the muscle come together. Steps 3 and 4 occur afterwards, are referred to excitation, contraction, coupling. You have to receive that nerve stimulation to excite the muscle so that then it can contract. So this is broken down into different phases. Phase 1 is when the motor neuron is going to stimulate that muscle fiber. And then phase 2 is the excitation, contraction, coupling. We're going to go through this in detail. And then there is, those of you taking my class, there is another video that I do have posted. It's also on YouTube for muscle contraction, where I go through, I have this drawn step by step on a whiteboard. You have to excuse my lack of artistic ability, but where I've gone through it step by step. So an action potential is going to arrive at the axon terminal. That's at the end of the axon on the nerve. The neuromuscular junction is where the nerve and the muscle, they are not physically touching each other. There's a little tiny space between them, but it's where they're going to be able to communicate. You're going to have the release. ACH refers to acetylcholine. That is a neurotransmitter. It is stored in the neuron. It is going to be released from the neuron, and it is going to bind to the sarcolemma. Remember, now that's on the muscle. Your permeability of the sarcolemma changes for ions, meaning it's going to allow certain ions to enter into the muscle fiber, which normally cannot. When those muscle fibers or ions enter into the muscle fiber, it's going to cause a change in the charge, what we would refer to as the membrane voltage. It's going to change it from a negative inside the cell to a positive inside the cell. That's depolarization. You have just a small area where initially you've got this local depolarization. And that's what we call implant potential. It's just in a small area. It's not the whole cell. Initially, it's just a small area, and that's going to trigger other steps to happen, allowing more positive charge to enter the cell, and then you generate the action potential. That action potential is going to travel. Once it reaches a certain level, it's all or nothing. You reach a certain level, it's gone. You've got an action potential. It's going to travel across the entire cell surface, the entire sarcolemma. Well, remember, you've got those T-tubules. So when the action potential is coming along, it reaches a T-tubule. It doesn't jump across them. It goes down the T-tubule. And what's right next to the T-tubule? The terminal cisternae of the Sarcoplasmic Reticulum, which is where calcium is stored. And that action potential moving down is going to trigger the release of the calcium. You don't have muscle contraction yet. You're just trying to stimulate the muscle. Once the calcium is released into the sarcoplasm, it now binds to the troponin. And when it binds to the troponin, remember I said anytime you have binding, you're going to have a shape chain. It's going to pull that tropomyosin off of the active sites so that now those myosin heads, they can bind to the active. And now that's you finally at contraction. We're going to go through all of these steps in detail. I think I have it down to about 27 steps. So your skeletal muscles are going to be stimulated by somatic motor neurons. Motor neurons, it means it's just bringing the information from the central nervous system, which is the brain or the spinal cord, and it's bringing that down to the muscle. The neuron, the axon is an extension from the neuron. Each axon has little branches at the end of it. And where those branches end on a muscle fiber, that's where we call the neuromuscular junction. So you have up here, here's your neuron up here, and you have an action potential coming down. And the very end of the neuron is referred to as the axon terminal. So this is the axon terminal. So you're looking right here at this one enlarged. This is your muscle down here. Now, the neuron is coming right in at the muscle, but it's not physically attached to it. There's a little tiny space. We refer to this as the neuromuscular junction because you've got a neuron and a muscle, and it's where they're interacting. Now, the space between the neuron and the muscle is known as the synaptic cleft. You have to remember, things want to go from high concentration to low concentration. And that's important to remember that as we go through this muscle contraction. So first steps, you have an action potential that is coming down that axon. You had a stimulus previously. And you have a message coming from the central nervous system, from the brain, saying, you need to move your hand. So that electrical impulse is coming down that neuron. When it reaches the axon terminus, an action potential essentially is an electrical charge. So when it comes down to the end of this neuron, that change in that charge triggers the opening of some of these ion gates, which are very specific for what ion they will let in. Calcium, remember earlier I said in the cytoplasm, it's normally low. It's higher outside the cell. So as that action potential comes down and you open this calcium ion gate, it's a voltage gate, so it's going to respond to the change in the charge that's coming down. Normally these gates are closed. The charge comes down, it triggers the opening of it. If calcium is normally higher outside the cell than inside, then it's going to go from high to low. So it's going to flow in to the axon, into the end of that nerve. What does it do when it flows in? Well, at the axon terminus, you have these little vesicles that are filled with the neurotransmitter. In this case, it's acetylcholine. If the neuron is at a neuromuscular junction, it's going to be synapsing with a muscle. The neurotransmitter will be acetylcholine. There are other neurotransmitters, like if a nerve stimulates a nerve, it may be different. But if it's a nerve to muscle, it's acetylcholine. So when calcium comes in, it triggers these vesicles containing the acetylcholine. Those vesicles are going to move towards the membrane and fuse with it, and when they do, they're spitting out, basically they're dumping out that neurotransmitter into the synaptic cleft area here, that little space. Well, if you have a high concentration here and you dump it into the space where there's none, goes from high to low concentration, and that neurotransmitter is going to diffuse across the synaptic cleft. Well, guess what's over here on the muscle side? You have these receptors that are very specific for acetylcholine, and the acetylcholine is going to bind. I don't know if you can see right here, you have those little green acetylcholines attached, and also right here and here. They are going to attach to these specific receptors. And whenever you have two things bind together, you're going to see a shape change. So this is what is explaining here, what we just saw, that axon terminus and the muscle are separated by the synaptic cleft. The acetylcholine, which is the neurotransmitter, is stored in those vesicles. On the muscle side, you have the acetylcholine receptors. So now, once your acetylcholine has bound to the receptors on the muscle side, so it diffused across, it has bound, so here we have this area now enlarged showing that it has bound. This is a type of a gate that responds to a chemical binding to it. And when that neurotransmitter binds to it, it opens. And this one is specific for allowing sodium in. Some potassium will leak out, but mostly you have sodium coming in because sodium is higher outside the cell than inside the cell. So it goes, once again, high to low. So the sodium is going to flow in. So you have this influx of sodium. Sodium has a positive charge to it. Keep that in mind. So this, once again, you've got the nerve impulse arriving, the acetylcholine diffuses across, binds, opens the gates to allow the influx of sodium. Now the acetylcholine eventually is going to be broken down by an enzyme called acetylcholine esterase, and that ultimately will stop the contractions. So this is just showing here when that enzyme comes in and it breaks it down, that will then close the gate because you've pulled the acetylcholine off. That closes the gate, stops the influx of sodium. There are a lot of different drugs and diseases that can interfere with events of the neuromuscular junction. Some of these are autoimmune diseases. Some of them are different toxins that can interfere with those steps. The resting muscle cell does have a charge to it, so we say that it is polarized. The inside of the cell, naturally at rest, is slightly negative. So when you have an action potential arrive from that neuron, you're changing the electrical charge inside the cell, and there's a few steps that are involved with this. So the end plate potential, this is going to be when you change the charge just in a very small localized region of a cell. So we're talking about a very small area. So that acetylcholine that was released, it bound to the receptors on the sarcolemma, and that caused those gates to open. I said it's a chemical gate because it's responding. It's also can be called a ligand because the way you open it is to bind a chemical, in this case acetylcholine. That sodium that has a plus charge to it rushed in. Some potassium may move out, but it's mostly sodium influx. That's bringing in a positive charge. Now, the inside of the cell, I said, is normally negative at rest. So if you're normally negative and you've got all this plus charge coming in, you're becoming more positive. And that's what depolarization is when you're going from a negative charge and you're getting closer to zero. You're becoming more positive. Because it's just in a small area, we call it the end plate potential. So that sodium's coming in here is just in this area right here that you see that change. Now, if the end plate potential causes enough change that it reaches a certain level that we call threshold, then what happens is it triggers the opening of what we call voltage gate sodium channels. Voltage gates respond not to the binding of a chemical, but they're going to respond to a change in electrical charge. So if you just have a little bit of sodium coming in and very quickly the acetylcholine is broken down and those gates are shut down very, very quickly, then you may not have an action potential generate. You just have that end plate potential, a little change, but it doesn't reach threshold, so nothing happens. You're not going to see a muscle contraction. But if you have enough influx of sodium coming in that it does trigger all this positive charge coming in and you reach a threshold where now it triggers the opening of those voltage gates, they open, allowing even more sodium to come in. Now you're going to reach threshold because now you've got a ton of sodium flowing in. You reach threshold. Once you reach threshold level, it's all or nothing. Boom, you've got an action potential. An action potential is where you're changing the electrical charge and it's going to travel across the entire length of the cell, not just a localized area like an end plate potential. An action potential is like, boom, you got the charge, it's gone. And it will travel the entire length of the cell. You definitely are going to trigger depolarization in this case. So once again, here's the localized area, the wave of depolarization. If it's just a little bit and it dies out here, nothing happens. But if it's enough that it's going to come and it's going to reach these voltage gates here, then that, notice right down here, this is negative. The charge is not there yet. But you've got this action potential coming. And boom, you're going to open these voltage gates. You have this huge influx of sodium coming in. Your potassium gates right now, notice these are closed. Potassium is higher inside the cell, but this gate's closed, so it's not able to flow out. So you're keeping your positive charge in and you're adding even more positive charge. That's why the inside of the cell now is becoming positive. This action potential is going to extend all the way down the length of the cell. Repolarization. This is the process where you try to go back and restore to resting conditions. How does it do this? Your sodium voltage gates close, and then your voltage gates for those potassium lines that we just saw will open. Well, if potassium is normally higher inside the cell and you open the potassium gates, then that means the potassium is going to flow out. So now you have potassium, this efflux out. You have the positive charge flowing out. That's going to make the inside of the cell more negative. A refractory period refers to the time period when the muscle fiber cannot be stimulated until repolarization is complete. You have to get everything restored back to the resting state. Until you do that, the muscle will not respond to any other stimulus. How do you help restore things to the resting state? Well, you had all that sodium that came in, and so you've got to get it out. The sodium-potassium pump is going to pump 3 sodiums out and 2 potassiums in. You do not want the sodium and the potassium to reach equilibrium on either side of that sarcolemma, because then you won't have a charge. You won't have a negative charge inside. If you do not maintain this electrical balance, that means you have no resting potential. If you reach equilibrium and you open up a gate, is sodium going to flow in? Not if it's at equilibrium. So you need to keep sodium higher outside the cell, so when you open the door, it's going to flow in to generate that action potential. If it's at equilibrium, it's not going to flow in. You never generate an action potential. And why is that so important? If you don't generate an action potential, you will never have muscle contraction. If you never have muscle contraction, and that's going to involve nerve impulses as well. If you never have nerve impulses and you never have muscle contractions, you will die. So you need to keep this in balance. It's one of the times when you don't want equilibrium to be reached. You need to keep this in balance, and the sodium-potassium pump maintains that change. It keeps the sodium out, because we do have some leak gates where potassium is trying to leak out, some sodium is trying to leak in. So it's kind of like, okay, you leak in, we're going to pump you back out. Come in the front door, I'm moving you out the back door. So the sodium-potassium pump, both sodium and potassium have a +1 charge. If you are moving 3 sodiums out and only 2 potassiums in, so 3 plus charge out but only 2 plus charge in, that also helps to maintain that the inside of the cell is negative and the outside is positive, which has to be that way in order for any of this to work. So this is shown with repolarization. The sodium channel here has been closed, but the potassium is open temporarily to restore. This is resting with inside negative, the outside positive. If you look at a graph, we can measure what the resting state of a cell is. And this is measured in millivolts. So you're down here, you get that impulse, and initially your sodium gates are going to open right along the neuromuscular junction. You get that implant potential. It's traveling along, now it reaches those sodium voltage gates, which open. Boom, you have all that influx, you've got an action potential going. All that sodium's flowing in, your sodium gates are open up here. So this is depolarization, you're going more positive. Right around here at -55 is threshold. So you reach threshold, you're gone. Now you're going to close those sodium gates. Close the door. Sodium's no longer coming in. Now the potassium opens, so your plus charge is flowing out of the cell. This is repolarization. You're trying to get back to resting state. Potassium's flowing out. Close those potassium gates. Now sometimes you kind of overshoot a little bit. And then now you're back to resting state. During this time period here, you cannot respond to another stimulus until you restore back to that resting state. The excitation contraction coupling events, or EC events. This is where the action potential now is moving along the sarcolemma, and we haven't gotten to muscle contraction yet. We just have the action potential that's moving along. It gets to the T-tibule. It's going to move down the T-tibule. Now you have some voltage proteins there along the T-tibules that are going to basically stimulate the release of calcium from that sarcoplasmic reticulum. So here's that neuromuscular junction where we had all those previous events. Your acetylcholine has bound to the receptors, opened the sodium gates. You got the implant potential. It moves along. Oh, now it hits the voltage gates. More influx of sodium. It's pouring in. You've got an action potential because you reach threshold. Boom. Action potential is traveling. Now it's going in both directions. Keep in mind. Oh, it hits a T-tibule. Now it's moving down here because it's going to move down here and then back up and move down here and back up. And it's traveling. This is the T-tibule. Here's your terminal cistern, which is where, if we look at the enlarged picture here, so here comes that action potential. It's coming down. It triggers the opening of these gates in the cistern. That's where you're storing a cistern, like a water cistern where you store water. You're storing your calcium in there. That voltage comes down. It opens the gate for the calcium to be released. Where is the calcium going to be released? Into the sarcoplasm, which is the cytoplasm of a muscle cell. Why does it flow there? Because it's normally not in there. So it's very high concentration in the cistern, low concentration here. So from high to low. Start to realize now why it's so important to understand diffusion. Things want to go from high to low. So calcium is flowing out. What happened to the action potential? Oh, it's moving on. This calcium flows out. What does it do? It's going to bind to the troponin, the yellow figure there. Remember what I said the troponin does. Calcium binds to it. When calcium binds to troponin, you get, surprise, you're going to get, whenever two things bind together, a shape change, conformational change. It's going to shift the tropomyosin. Where has the tropomyosin been? That's that strand that's been lying over the active sites. So when calcium binds to troponin, it triggers a shape change, moving that tropomyosin off of the active site. So now that active site is exposed for the myosin head, boom, to bind to it. So this is what all of this is saying, what I just went over. The nerve impulse travels down the axon to the motor neuron. Of the motor neuron, it reaches the axon terminus. Excuse me. Triggers the opening of calcium gates, allowing that influx of calcium. That triggers the release of the acetylcholine from the vesicles, which will diffuse across the synaptic cleft. They will bind to receptors on the sarcolemma, causing the opening of sodium gates, generating the implant potential. If it is strong enough, as we see here, just going to move along. If it is strong enough, it will then open the voltage sodium gates, allowing further influx of sodium as it moves, generating now an action potential which will move down the sarcolemma to the T-tibules, allowing the release of the calcium. So as I was saying, normally tropomyosin is blocking the active site, and calcium is very low. If the active site is blocked, myosin head cannot attach, and muscles nice and relaxed. When calcium levels increase, they will bind to the troponin, changes the shape, moves the tropomyosin off those binding sites, allowing the cross-bridging to occur. So once again, hopefully you can see how the calcium is binding. Now all of the active sites are exposed. So now we finally get to the actual contraction, because you haven't had muscle contraction yet. You have all this preliminary work, but you're not even at the muscle contraction yet. So there's 4 steps of the cross-bridge cycle, what we call. You have to attach that myosin head to the actin, to that active site. That's the cross-bridge formation. Then you have what's known as a working or a power stroke. That myosin head is going to pivot, and it's going to pull that actin filament towards the inline. So it attaches, and there's going to be a stroke. Think of like in a boat with oars, if you've ever been in a rowboat or a canoe or something. You put the paddle in the water, and then you have to pull it. Then you have cross-bridge detachment. This is going to involve ATP, and then the myosin head has to cock, kind of get it into position for the next stroke. So this is, in some ways, very much, if you've been in a rowboat, if you've been in a canoe, you have the paddle, you reach forward, you put the paddle in the water, and you pull. So that the pulling would be the power stroke. Lift the paddle up, that would be detachment. Now you've got to push the paddle forward again for your next stroke. Same thing with the myosin. It's got to recock that head. Recock the head, put it in the water, pull, lift up the paddle, reach forward. That's the same thing that the myosin head is doing. So right here, it attaches. It's going to pull so that it is sliding the blue or the thin filament, that actin. It's going to slide it. So it attached right here, and now it's going to slide it. Now it has to detach from it. That's going to require ATP, and then recock so it can bind to the next site. Bind to it, pull. So if you think of it, if you were to look ahead, so if it initially bound here and pulled, released, attach, pull, release, attach, pull, release, attach, it is sliding the blue filament from right to left in this case. From a clinical standpoint, what is rigor mortis? Well, 3 to 4 hours after death, the muscles stiffen up. The peak rigidity is about 12 hours after death. Why does this occur? Well, you have your intracellular levels of calcium have increased because there's no more ATP being synthesized because the animal is dead. The calcium cannot be pumped back into the sarcoplasmic reticulum. And so you're not synthesizing any more ATP. You need ATP in order to detach that crossbridge, in order to detach the myosin head. So because you don't have any ATP, the myosin head is staying attached to the actin, and so the muscle is staying in a state of contraction. There's no relaxation. It's going to stay very rigid like that until the muscle proteins start to break down, and eventually then the myosin is going to be released from the actin. So you need to be familiar with these steps of the muscle contraction. Once again, how does relaxation finally occur? Well, remember I said you have the acetylcholine esterase, which is breaking down the acetylcholine in that synaptic cleft. As it breaks it down, it does close that sodium gate. And then you restore to the resting state. How? Because you have the sodium. The sodium gates close, the potassium gates open, allowing the potassium to be moving out of the cell, restoring back to that negative charge. That, combined with your sodium-potassium pump, does restore the inside of the cell back to that overall negative charge. That is also going to close those gates, and the calcium is going to basically go back into the sarcoplasmic reticulum for storage again. And so when it is released, when calcium is released from the troponin, it is going to then move the tropomyosin and cover the active sites again. So it's no longer available for the myosin heads to bind. And that's when you have then the relaxation occur. You do need to be familiar with this process. And something else to keep in mind before we continue on is that obviously you're seeing how calcium plays a role in the neuron at the very beginning for the release of acetylcholine, and then plays a role at the end to actually allow for the contraction to occur. Sodium plays a role, potassium plays a role, and hopefully you're starting to realize why the body has to very carefully maintain concentrations, appropriate levels. You don't want too high or too low of levels of some of these ions, such as the calcium, sodium, potassium. And the body does indeed very closely regulate the amounts. Usually it's monitoring it in the blood. Too low of concentration or too high can have some very serious ramifications on the nervous system, on the muscular system, because it can interfere with all of this. And so hopefully you're starting to see how, number one, systems work together, homeostasis that we talked about in the very beginning of this course, how it's this trying to maintain this internal balance. I often describe A&P as kind of like this puzzle that we keep throwing pieces out at you, and hopefully you're putting the pieces together, and you're starting to see and visualize the overall picture now of what the puzzle's going to be. You start to see how everything works together. You can't have a puzzle without all of the border pieces in. You can't have a puzzle with, if you're like me, I like doing puzzles, and so frustrating when I was doing a 1,000-piece puzzle and there was one piece missing. Everything works together for the overall picture, which in this case is the overall healthy functioning human body. So hopefully you're starting to see why, well, maybe if somebody has an issue with muscles or an issue with their nervous system, are you starting to maybe understand why, well, maybe you need to look at the level of sodium intake that they have or the level of calcium intake that they have. If it's too high or too low, what ramifications will that bring? A&P, the purpose of this class is to teach you what is the normal functioning so that you can understand with different diseases and disorders how the interference is disrupting the normal patterns of the way things are supposed to go. You can't treat a disorder if you don't know what the normal functioning is, because how do you know if it's disrupted it? And everything has an effect. Everything is interrelated. When we look at whole muscle contractions, a lot of the same principles are going to apply. Contraction produces muscle tension, which is the force that's exerted on. Now, contraction may either shorten the muscle or it may not. Isometric contraction, there's no shortening. The muscle tension increases, but it doesn't exceed that load on it. Isotonic contraction is when the muscle does shorten because the muscle tension exceeds the load on it. Oftentimes, the force and duration of the contraction is going to vary depending on the response to the stimuli. How frequent is it? What's the intensity of the stimulus? Every muscle is served by at least one motor neuron, sometimes more. So how many stimuli are coming in? The motor unit is the nerve muscle functional unit, as we have said before. You've got the motor neuron, you've got all the muscle fibers So if you have only a few, say you have 4 fibers being stimulated, then you have a lot more fine-tuned control. But if you have 100, it's going to be typically a stronger contraction, but not quite as delicate in the fine control. And this is just showing how you can have the branching and how much is stimulated. A muscle twitch is a very simplest type of contraction. The muscle fiber has been stimulated by a single action potential from that motor neuron. The muscle fiber is going to contract. And remember, skeletal muscles, we're talking quick, over, done, relax. You can measure this. There's 3 phases of a muscle twitch. There's the latent period, there's the period of contraction, and then the period of relaxation. So the latent period is that period of early on when there's no muscle tension being seen. It's kind of all that phase leading up to contraction. Then you actually have the contraction where you actually have that crossbridge formation. And then the relaxation. That's when you're trying to restore back to the resting period. That's when the calcium is going back into the sarcoplasmic reticulum. Muscles will contract very quickly, and it takes a little longer time for it to really go back to its resting state. So here is your latent period in this darker blue right here. Here's the contraction period. You've got the cross-bridging occurring. So this is the acetylcholine's binding to the muscle. You've got the influx of sodium, depolarization occurring, that action potential is traveling down the sarcoplasm, all those events, but it's very quick. And the time here is milliseconds. And then, boom, here's your period of contraction. But to get back to that resting state, getting all that calcium back into the SR, that takes, everything is relative. It takes a longer time period to do that. That's your period of relaxation. Differences in the strength and the duration of twitches, that's due to different metabolic properties. Eye muscle contractions, very rapid, very brief, but some of your larger muscles, the contraction might be slower. It's able to hold it a little bit longer. And this is just showing how long the contraction period is. Some of your eye muscles, the gastrocnemius, it's a large muscle on the back of your thigh, and the soleus, etc. Muscle response to changes in stimuli frequency. A single stimulus results in a single contractile response, just a single muscle twitch. Now, if you can have what we call wave or temporal summation results when you have 2 stimuli that are received by the muscle in very rapid succession. So the muscle fibers, you're restoring back to that resting state. It's in that relaxation period. But it doesn't completely relax between the stimuli. So the twitches are going to increase in force with each stimulus because you've got more and more calcium. If you have 2 stimuli that are received by a muscle in rapid succession, you can get a very smooth, continuous contractions that add up. There's summation. It's a wave summation type of thing. And then what we call complete tetanus. This is where the stimuli frequency increases the muscle contraction. The tension reaches its maximum. You get like this one long, smooth, sustained. If you graph it out, it plateaus. Now that can lead to muscle fatigue. So here is just a single stimulus. Right here is the arrow of the stimulus. You have your contraction, relaxation, boom. This we have what we call incomplete tetanus. You have a stimulus here, the contraction. It's relaxing. Now you have another stimulus. And because you've restored enough, it can respond, but you're starting at a closer You don't have, if from a graph standpoint, you don't have this so much to go. So you gradually get stronger and stronger contractions. And then this is what's known as complete tetanus. As you can see, you reach this plateau. This muscle is going to fatigue. Recruitment just talks about having multiple motor units. You have a stimulus coming in. There's different types, subthreshold stimulus. That stimulus is not strong enough to generate that action potential, so you didn't reach threshold, so you don't see any contraction. Threshold stimulus, the stimulus is strong enough to cause the contraction because it did reach that stimulus. And then maximum stimulus is the strongest stimulus that you can receive. So down here, you just had a small little stimulus, but you did reach that threshold point. But you can see increased amount. So you wouldn't see any. You have to reach threshold in order to see a contraction. Muscle tone. This is where you have some muscles contracting, and anytime you do have some muscles contracting, not real strongly, just a slight contraction, and that allows what we call muscle tone. It keeps the muscles firm and healthy. They're ready to respond. It's due to spinal reflexes. Now, is it always the same muscle? No, they're constantly changing for good muscle tone. You need energy in order for muscle to contract. Where does this energy come from? Well, ATP, adenosine triphosphate. That is what you need in order to supply the energy that's needed for the muscle contraction. And why do you need it? Because you need it during that crossbridge. It will attach, but you need the ATP to detach it and recock the head so it can attach now to the next active site. You need ATP to pump calcium back into sarcoplasmic reticulum, and that sodium-potassium pump, because you're going against the gradient, you need ATP to run it. So the cell needs ATP. So for muscle contraction, you do have some ATP that is stored in the cell, but when you have a muscle contraction occurring, you're going to run out of that available ATP in about 4 to 6 seconds. So your muscle contraction, say you're running, you need to run more than 4 to 6 seconds. So where does the rest come from? There's several different pathways, and these are the 3 most common. You can get more ATP from breaking down creatine phosphate. You can get some ATP from anaerobic pathways, and then finally from aerobic respiration. Anaerobic means without oxygen. Aerobic is with oxygen. So you need, I always say, if you saw me running, number one, I don't run, so if you see me running, pass me up, because there's something going on, and it would probably be in your best interest to start running too, and just pass me up. So what would I need? Well, first off, if I started running, the first roughly 5 seconds, I would use up any available ATP that I had in the cell. Now what I do, well, creatine phosphate is a molecule that's in the muscle fibers, and so it can be broken down, and it will generate creatine and ATP. So it's another source. However, it's only going to last for about another 15 seconds. Still not enough to help out a whole lot. This is just showing the breakdown process for it. So the muscle still needs ATP. So now what happens? Well, now it's going to switch to the anaerobic pathway, because in order to produce ATP, you need oxygen. You have probably used up the oxygen that was necessary at this point. And so glycolysis is the pathway of breaking glucose, which is a sugar, down. And all cells can break glucose down. All cells carry out glycolysis. Glycolysis itself does not require oxygen, and it will produce 2 ATPs and then pyruvic acid. Well, that's great, 2 ATPs. That's not going to do a whole lot, but we'll take whatever we can get. If oxygen is not present, then the pyruvic acid will move into, it's kind of like a fork in the road. You either go right or you go left. If oxygen is not present, then it has to go the anaerobic pathway, which is fermentation. If oxygen is present, then it will go the aerobic pathway, which will generate a whole lot more ATP. It's much more efficient. But if it's not there, you can't do anything with it. So for the fermentation pathway, very broadly, fermentation, you either have alcohol fermentation or acid fermentation. And as a microbiologist, yes, I can tell you there are a lot of microorganisms that will carry out the alcohol fermentation where the final product is some form of alcohol, of which we have learned to really capitalize upon that. We know what organism with which type of grain gives us what alcoholic beverage. Humans do not carry out alcohol fermentation, which is probably a very good thing. Instead, we carry out acid fermentation, and specifically the acid that we produce is lactic acid. So when oxygen is not present, glucose is broken down to pyruvic acid no matter what. That pyruvic acid, when oxygen is not present, will then be broken down into lactic acid. That will still allow you to produce some ATP. Only 2, but hey, 2 is better than nothing. So here's what's happening. Your glucose, it's being broken down. You're getting some ATP, a little bit better than nothing. And what are you producing? You are producing lactic acid. And remember, this is without oxygen. Okay, well, the problem with this, it sustains you for a little bit, but not very long. The other problem is the product that you're producing is lactic acid. It is an acid, which means it's going to drop the pH. Your body very closely monitors your pH. Your pH in your blood needs to be between 7.35 and 7.45. And if you start dropping that, there are some severe ramifications. Your body lets you know that it does not like that lactic acid that you're generating. And that's why you start to feel that burn, that achiness. That is the buildup of the lactic acid. Your body's saying, I don't like you right now. I don't like that you are doing this. Maybe you should stop running. If you work out a lot, you have built up a tolerance. You can tolerate much higher levels than, say, someone like me who doesn't work out. That will get you some time. Now, aerobic respiration, this is much more efficient. You take that glucose and you break it down into, by aerobic respiration, ultimately you're going to break that glucose down to carbon dioxide, water, and generate a lot of ATP. And that is what you're going to need. But you need oxygen. So think about this. Is this all starting to make sense? When, even if you're just, say, walking or hiking or running, whatever it is that you enjoy doing. You've all been in situations where maybe you're trying to rush to catch up to a friend or catch a bus or whatever it is that you're doing. You're stationary. You start moving. Those muscles need ATP. The available ATP runs out in about 5 seconds. About 15 seconds after that, it has run out of the creatine that it was able, the creatine phosphate, break it down, produce ATP from that, After about 15 seconds, you've run out of that. You need oxygen to make ATP. So as you're running, you have run out now of the oxygen to make the ATP in the muscles. So you switch over to anaerobic. It's enough to produce a little bit of ATP to keep those muscles going. What happens when you exercise? Your respiration rate tends to increase, doesn't it? You breathe faster, you breathe deeper. Why? To get more oxygen in. Your heart rate goes up. Why? Your heart starts beating faster, your blood pressure goes up typically, so that you need to increase the blood flow to get the oxygen that you've been inhaling. You have got to get it to the skeletal muscles. That's why you've increased your respiration rate to increase the oxygen levels. You increase the blood rate, blood pressure to get that blood flowing faster to the muscles to bring that oxygen there that is needed. Because the oxygen is needed for the aerobic respiration to produce the ATP that is needed in order for the muscle contraction to occur. Now, that lactic acid that was produced during the fermentation or the anaerobic respiration, that is a reversible reaction. So once you get oxygen, that lactic acid over time can be converted back to pyruvic acid, which then can flow to the aerobic respiration and continue on the process. It just takes a little bit of time. So this is showing in here with aerobic pathway, you've got glucose making the pyruvic acid. Now that you have oxygen present, it will go through the aerobic respiration. Given a choice, if oxygen is present, it says forget that fermentation, we're going through aerobic respiration because it's much more efficient. We generate, instead of 2, we will get about 32 ATPs. Fatty acids and amino acids can also feed in various steps along the way here. So during sports, aerobic endurance versus anaerobic threshold, at what point does the muscle the metabolism convert to those anaerobic pathways or not? So this is just kind of showing first you're using the ATP that was stored, now the creatine phosphate. Now you've used up the oxygen. So, if you're like, this, okay, this would be me doubled over going, oh my goodness. And, you use an anaerobic if for prolonged, you've increased respiration, increased heart rate, so that you can generate more and more ATP. Muscle fatigue. Physiologically, you're unable to contract, even though there's stimulation. It's tired. Why does this happen? Usually there's an imbalance of the ions. The calcium, the potassium, phosphorus levels are off. Prolonged exercise can also damage the sarcoplasmic reticulum. It can interfere with how the calcium can be released and regulated. So it's usually due to an ion imbalance with muscle fatigue. It's not usually due to the lack of ATP. For a muscle to return to its pre-exercise state, you have got to restore the oxygen reserves. You've got to get that lactic acid reconverted back to pyruvic acid. Glycogen, which is a storage form of glucose, has got to be replaced. You need to replace that free ATP in the creatine phosphate. So you've got all these things. You have to restore everything back to how it was. And that's going to require some extra oxygen to get everything back in place, which is why once you stop running, you don't just automatically stop your deep breathing. You're still trying to get enough oxygen to help restore everything to that resting state. Years ago, that's what we used to call oxygen debt. Now we give it the name excess post-exercise oxygen consumption.